Today we have Tracy Heath visiting us, but before that, there's a couple of general announcements. So just to remind you that today at 4 p.m., Alison Schultz, who is an assistant curator of ornithology at the Natural History Museum here in LA, she's going to give a talk on evolution across timescales, comparative and population genomic studies of host pathogen coevolution in birds. That talk is going to be at 4 p.m. in the La Cretz Hall, third floor. And also want to remind you that at 4:30, we're going to have a happy hour for Tracy. That's going to be at the Botanical Garden at the La Cretz Pavilion. So you can go to Alison's talk if you want to, and then run to the garden. This is open to anyone. You can just go home, bring your kids, bring your family. Hopefully, we're going to have a good time talking to Tracy. So if you sign up to talk to Tracy today and you want to talk to her, that's a good chance. And I think that's what I have. So now, it's for me an honor to introduce our speaker today, Tracy Heath. Tracy is an assistant professor in the Department of Ecology and Evolutionary and Organismal Biology at Iowa State, where she has been there for 4 years already. 4 years. Tracy is a computational biologist, theoretical computational phylogeneticist, but she also does some empirical work, and she maybe touches a little bit on that today. But just a little bit of her background: she got her BA in Boston University, where she was actually later on the DNA sequencing facility manager. So actually, she started as an empirical biologist doing empirical biology in a lab, sequencing genes, dealing with molecular data, and just after her, when she started her PhD, wasn't when she transitioned into becoming a hardcore computational biologist. So for those of you who are sort of worried that she's a computer scientist, she's not. She's a pure biologist. She knows how data looks, and she became one of the leaders in computational phylogenetics right now. So after her PhD at UT Austin, Tracy moved to University of Kansas and Berkeley, UC Berkeley for a postdoc, and that's actually where we met. We were postdocs in Berkeley. And Tracy has been one of the leaders in using Bayesian statistics for phylogenetic inference, but also for modeling evolutionary processes. For those of you who are here who have been using phylogenetic methods, maybe you remember the program called Mr. Bayes. It's one of the programs that people use for Bayesian inference of phylogenies. And Tracy was, or is, one of the core members of the team who developed Mr. Bayes. But that program is sort of getting revamped by a new program called Red Bayes. That is what Tracy is going to talk about us today. I just want to say before she starts that she has been super successful funding. She has an amazing lab these days, but also something that I really admire from Tracy is that she's been one of the strongest advocates for promotion of diversity and inclusion in STEM, particularly in evolutionary biology, and specifically in computational phylogenetics, where she's been supporting many people. She's invited to panels. She teaches in different workshops across the world, and she's one of our sort of leaders in promoting these, but also one of the leaders in the field. So with that, I just let Tracy get going. Thank you.
All right, thank you. Thank you for the introduction, Felipe. That was awesome. Can you hear me okay? Am I heard okay on this? Okay. Thank you for inviting me to give this talk. It was sort of fortuitous. I'm actually also giving a talk on Friday at the Rancho Santa Ana Botanic Garden. And so it gave me an opportunity when I realized I don't want to give the same talk 2 days apart. I'm going to write a new talk. And so I made a new talk. So it's the first time I'm giving this talk. So I'm prefacing this because it might be a disaster. But I'm kind of excited about like sort of the directions that we're going with the software that we're working on. And a lot of people have asked me about like updates on like, what Rev Bayes is and how it's working and what we're planning on doing and what it does and where it came from. And so that's kind of the talk that I put together today. And so if you're interested, if you came and wanted to hear about fossilized birth death stuff, I'm actually going to talk about that at the Rancho, and so but it'll come up here too. Anyway, so just to start off though, I kind of want to give a little bit of an overview about my perspective in biology and what we're working on in my lab. So there's kind of really three major components of my research in my group. And so one of the projects that I'm working on now is looking at co-diversification and patterns of coevolution in this, the fig-wasp mutualism. So we're working on sampling species from Panama and within the community of figs and wasps that are in Panama, and looking at patterns of macroevolutionary patterns of co-diversification. And so this is work that's really led by two collaborators, John Nason and Alan Harry, and we also have a postdoc, Jordan Sattler. He's about to submit a manuscript like tomorrow, describing a lot of the sort of really interesting patterns of host switching and co-phylogenetics that we see in this system. And this is also work that's contributed by my student Kevin and my other grad student Wade. And so this is a cool video that Kevin took of fig wasps leaving a fig fruit, and yeah, I just think it's so cool. It's one of the coolest systems in the world. And so this is like kind of underscores like the biological motivation for a lot of the work that I do. So when we started collaborating on this project, I was really excited about co-diversification and understanding this in a phylogenetic context, but like didn't really understand the biological systems to which one would apply this. And so this collaboration really kind of spurred like a lot of future directions in my group about thinking about these problems, because I think there is a kind of a real open niche in phylogenetics for doing co-diversification. So the other main component of my research is developing statistical methods for integrating fossils into phylogenetic analyses of extant taxa. And so this is probably what most people, if you're familiar with things that I've done, know about. And so a lot of this really centers around the theoretical aspects of extending and developing new process-based models on describing how we sample the fossil record and also how diversification occurs back in time. And so primarily in my lab, my postdoc Walker Pett is involved in this project. But within this group, like because we're developing these new methods, we've also been able to attract people who are applying these methods to empirical systems. And so I have two postdocs, one of whom some of you might recognize. So Janet Buckner is a NSF postdoctoral fellow working on the macroevolution of all answer forms, so waterfowl. And Dagmara Joash is has a Marie Curie, and she's really interested in using these same methods to actually date, difficult to date formations in the fossil record. And so taking empirical systems and kind of, has really kind of informed us in ways in which we need to extend and develop these methods for, doing better phylogenetics with fossils. And so both of those projects in my lab are kind of enveloped by like the main way in which we do our work. And so, for many people who do empirical analysis, they like have their lab and they do sequencing or genomics or something is really how you get to answer the questions that you want to answer. For us, we are like all of the new stuff that we're doing is kind of sitting within this like framework of developing a piece of software, and all of the software that we're working on is intended to be a mechanism for other researchers to access the methods that we're developing. And so three members of my group, so Walker Pett, Wade Dismukes, and Josh Justison are involved in this research. But this is, like all of my research is heavily collaborative. And so this really comes, together because I have a lot of other people who, I'm working with a lot of other people who are contributing to this project. And so, when thinking about what to talk about here, like I really wanted, like I'm starting to get excited about the directions of the software that we're working on, and so that's kind of motivated me to focus on this part of my work for the purposes of this talk. So for today, I'm really just gonna talk a lot about RevBase, what it is, sort of where it came from and what it does, but also kind of the philosophy and theory behind how it's engineered. And that really gets it describe how we describe statistical models in RevBase and in hierarchical Bayesian modeling generally. And so those are the things I'm going to focus on. So RevBase is a fully integrative Bayesian inference package that uses this idea of probabilistic graphical models, so a way of describing a model, and it's interfaced with an interpreted programming language. And so I'll talk about these in more detail. We released a paper for RevBase in 2016, and that's the citation. And obviously, here's where you can find the software. So a little bit of history of RevBase, and many of you might be familiar with RevBase's successor, Mr. Bayes. And so Mr. Bayes was developed and created by John Holzenbeck in about 1999, and he really created this program like to do some research that he was interested in, and he called the C file in his computer Mr. Bayes because he didn't know what to call it. So when he published a paper and people emailed him and asked him for what he wrote, he sent them this program, and they thought it was called Mr. Bayes, and that's what it became, right? So then this became really John's like research, environment, and that's what he worked on until 2000, when Fredrik Ronquist from the University of Sweden started working with John and co-developing Mr. Bayes in about 2000. And so, as the development of Mr. Bayes extended, and as the field matured for Bayesian phylogenetics and model-based phylogenetics, a lot of more and more complex models for describing molecular evolution and macroevolution were being proposed. And so in that time, both Fred and John realized that there were some significant limitations with Mr. Bayes and how it was designed that limited how you can extend it and develop new models and put new methods and models in it. And so they decided that they needed to completely rewrite Mr. Bayes, and also to rewrite it, instead of in C, to write it in C++, which would enable some more flexibility with the much better object-oriented programming language. And so that's when they started a new rewrite and called it Mr. Bayes 4, which was in C++. And so in the process of doing this, they realized there are some other problems. There's one of the biggest challenges is how we actually describe models when we're implementing them in a method. How do we tell POP or Mr. Bayes or anything else what model we want to use? We're really limited by the vocabulary of these file formats that we've created, and they recognize this at this stage as well. And at the same time, John actually started interacting with Michael Jordan at UC Berkeley, who's a computer scientist, who informed him about the whole wide field of machine learning that uses what's called probabilistic graphical modeling to describe statistical models. And so this really inspired Fred and John to pursue this as a philosophy, or at least a inspiration to work on the design of this new rewrite for Mr. Bayes. But then they realized that so much was changing between Mr. Bayes and RevBase that they kind of needed a new name, and so that's why they picked RevBase. And so this also so RevBase is there to also mark the fact that it's different from Mr. Bayes, even though Mr. Bayes is its sort of ancestor. There is still some code from Mr. Bayes that's in RevBase, but it's also like to acknowledge the fact that Thomas Bayes was a reverend and honor the software with his actual title. So this was about in 2006 where the project name shifted and the incorporation of this graphical model framework came in, and they also realized that they needed this new model specification language. And so they began to work on that with some collaborators who were involved in the project at that time, and then over the next 3 years, worked on this conceptualization of what was eventually to become Rev Bayes. So in about 2009, in 2009, Sebastian Hunne joined Frederick Ronquist's lab as a PhD student, and so he became heavily involved in the development of Rev Bayes, and they really began and sort of originated the Rev Bayes code base in about that time. And so this is actually a graph of sometime in August or October of 2009 of the Rev Bayes code base, and you can see a couple people in there. So there's little avatars for the developers, and so those are John and Fred and Sebastian. And so when Sebastian became involved, because Sebastian's background previously had been in computer science, so he helped motivate and work on the software, like the engineering of the software quite a lot. So then from 2010 to 2011, in this time, Sebastian actually really took over the project and established himself as the leader of this development, which was excellent because both Fred and John had a lot more responsibilities than he did. And then the project also began to expand to include a lot more collaborators. And so this is actually when I became involved, but also some of the other core developers as well. And then John had some contest for this logo, which most of us actually hate, but we can't get rid of it. So then from about 2010 to 2014, a lot of Rev Bayes was changing. A lot of us were involved in the development. The code base expanded quite a lot. We had a hackathon in France, and things started to really solidify and become more and more stable. And this is when it became actually really quite exciting to be part of this project at this stage, because then you could start to see the potential, like, oh, here's what I can do. And like once I started really implementing things in it, I was like, oh, it's easy because everything's in place for me. And so this was really a fun time for this project. So then in 2014, we had a workshop at Nescent, where this is when we kind of like put Rev Bayes out there for people to actually use, and it was we had to do a lot of work leading up to it, and then also realize like after the workshop was over, like this thing actually works. So that was awesome. And then in 2016, we officially released Rev Bayes and published this paper that was in Systematic Biology. So basically from now on, Rev Bayes is a huge collaboratively developed software project, which involves quite a lot of people. This is our last hackathon in France, where we had a subset of us who were there. There's a panorama. And so, we have over 30 contributors. We have, Rev Bayes is featured in, or we have standalone Rev Bayes workshops, about 3 to 4, 5, 6 of them a year, depending on how many of us what our other obligations are. And we have many, many new models and methods and lots of other things to do. And so that's kind of where we are now, kind of looking forward to the project. In this last hackathon, was talking to the group, I was like, well, what do we think Rev Bayes 2.0 looks like? And so we're starting to discuss what does it mean to go from 1.0 to 2.0. So for anyone who develops software, that's kind of a big deal. And you can acknowledge that what is the major transition between these major version numbers. So at this stage now, Rev Bayes has sort of the lead developers, and really Sebastian is doing much of the project management at this point. So Sebastian has just started a new faculty job at the University of Munich. And then there's several core developers, and this is just really kind of a subset of us, but kind of with Michael, myself, and Bestien, kind of having been with the project since 2011, and then postdocs that have been working at various stages on the project are heavily involved still today. And so, we also, like as a collaborative and open source software project, we also have this open to anyone who really wants to develop in RevBase. And so we have contributions from over 30 people, but pretty solid ongoing work from at least half of those. So there's quite a lot that keeps being added to the project, and it keeps growing, and it's impresses me very frequently with how many things it can do. So within our project, if you just look at the core code, there's quite a lot of code. We have it's over 200,000 lines of code and many commits. We're starting to work on and improve our documentation quite a bit. We have a website, revbase.com, where we have all of this documentation with many extensive sort of recipe-based user tutorials. We also have tutorials for any new developers who want to become involved in the project that kind of outlines the structure of the code and how it's designed. And all of our workshop materials are posted on this site. So we are also thinking about ways of interfacing RevBase with either a user implementing an analysis, or on the back end how you might visualize and process output. So we have a visualization package called RevGadgets. That is for you're looking at the output in R and doing some nice things, ways of reconstructing ancestral character states on a tree and visualizing those in nice ways using a lot of the machinery that already exists in R. We also have several interfaces that we're currently working on. So we recently got funding from an NSF ABI grant that's motivating, increasing our user base by improving these interfaces and thinking about how RevBase fits within this paradigm of research reproducibility. So the interfaces that we currently have and are working on, so we have an interpreted language. So language is how you interface with the program and define a model and describe it and do your analysis. And this language actually is inspired quite a lot by R and the model specification language in RevBase. So we hope that there are aspects of it that are kind of familiar to people and easy to use and learn. That said, with the interpreted language, just like with anyone getting started for the first time in R, you recognize that there's a significant learning curve, right? So, this can be pretty hard when you don't have familiarity with it. And so, this implores us to think of newer ways that we can at least get people introduced to the software so that they can use it in some easy ways. And so we have a Jupyter Notebook kernel, so this will allow you to kind of write extensive notebooks in using the Jupyter framework. And then we have an interactive GUI. And so the GUI itself is not quite released yet, but this is kind of a new way of doing in the same way that RevBase is flexible and modular, like the GUI is also flexible and modular. So it kind of matches a lot of the philosophy behind RevBase. So on the website you can find lots of this information. So for anyone interested in a description of RevBase and other things that are available, there are actually jobs listed for postdocs and PhD students because people in the project are recruiting. And then if you go to the tutorials page on that website, you'll see this extensive list of user tutorials. And so this is just kind of a subset of some of the things that you can do in RevBase. So we have introductory tutorials that explain, use RevBase to explain MCMC. We have tutorials that take the user through writing Metropolis Hastings moves in the Rev language. and also basic MCMC and molecular phylogenetics. We have tutorials detailing all aspects of phylogeny inference with different data partitions or substitution models. Within the divergence time estimation, we have different ways of dating phylogenies. We also have different approaches for estimating diversification dynamics of phylogenies. We can do gene tree, species tree estimation, and also biogeographic analysis under the DEC and epoch models and other things like that. We have process-based models for dating phylogenies with biogeographic, paleogeographic data. We can do look at chromosome evolution and how things like chromosome evolution might be correlated with rates of diversification. Discrete morphology and continuous trait evolution, so lots of aspects of looking at continuous characters on trees, and then various methods, so MCMC diagnostics, model selection, and model adequacy. And importantly, there are a lot of these different approaches and methods that you can do in RevBase, but the nature of RevBase makes it so that you can actually do a lot of these jointly at the same time. And so because you can do them jointly, if you're doing a biogeographic analysis, you can do that while you're co-estimating your phylogeny and accounting for uncertainty in the tree and the branching times while simultaneously while estimating your ancestral areas. And so this is really what even though there's a lot you can do in RevBase, the concept behind the software is and sort of hierarchical Bayesian modeling generally is that you can do this integrative Bayesian inference, which is can more appropriately capture our statistical uncertainty in various phylogenetic parameters. And so that really is like kind of the underlying philosophy of this project. So why do we need something like RevBase? It's, there actually are quite a lot of nice Bayesian phylogenetic programs out there. But, for those of us who are developing in things like Mr. Bayes, we run up against some significant challenges. So the issue with these pieces of software is that they just aren't flexible enough to start accommodating the new methods and new complex models that are coming online these days, and that we're thinking of and wanting to put into a piece of software and use to test and validate and assess with simulations. And so John actually really likes this analogy of software written for, like, in this sort of same way as you would have like a control panel in an airplane or a shuttle, where it's just a bunch of toggle switches. So if you think about Mr. Bayes or POP, like to go and do an analysis in POP, you open it up and you toggle on and off things that you want to do. And so in the GUI version of that software, you actually have to find space and real estate for all of these switches. So that's difficult, right? What would make sense is because a lot of the ways that we actually design these models is like really a series of building blocks, right? We have your model for sequence evolution, your model for how the rates vary across your alignment, a model for how your tree, like that you get your tree from. All of those are components of a bigger model, and they're just building blocks. And so what we would rather have is a set of, basically your toy box full of those blocks that you can then assemble to be the model that you want it to be. And so this was really kind of what motivated this. And within statistical modeling, things like wind bugs were already developed for these types of problems, except that none of those pieces of software were at the point where they could even accommodate something like a phylogeny. And so this is what kind of was really the underlying motivation for the shift toward this really flexible framework. So to kind of get at that more specifically, so if anyone who's used Mr. Bayes, if you open the program and look at the default model settings, so these are the default settings for all of the priors on the model parameters, you get this list here. And so if you're doing a phylogenetic analysis, so in a Bayesian framework, we have priors, and those priors are meant to capture our a priori statistical or understanding of this sort of distribution or probability of certain parameters, right? Those things should capture our uncertainty in the values of those parameters. And say I was setting up a model and I happen to have really strong a priori knowledge of what the shape parameter of the gamma distribution that describes how rates vary across the alignment. And perhaps I decided that I thought that parameter should really be distributed according to a gamma distribution, or some other distribution that might not be quite as confusing. But if say I wanted to put a prior on this parameter that was a gamma distribution, and I went to try and do that in Mr. Bayes, I would see that my only options are a uniform distribution on that parameter, where I could only it would have uniform probability between some minimum and maximum value that I set, an exponential distribution, or I could fix it at some value. Those are my only options. The other option would be to go into the core C code of Mr. Bayes, implement a new prior for this parameter as a gamma distribution, and then also implement the Markov chain Monte Carlo algorithms for proposing new states under that prior and accepting or rejecting them. So, if you don't happen to be very familiar with programming in C, or the theory behind Markov chain Monte Carlo, it might be actually really difficult to do this. And to do that for something as simple as defining a standard prior distribution on a continuous parameter. And so this really is the sort of toggle switch kind of idea that you have when you develop statistical software this way. So a lot of software projects in phylogenetics are moving toward a more modular approach. And so these types of packages, the benefit here is that we don't have to, recode the same thing over and over and over again. So in Mr. Bayes, this has an exponential prior on it, this has an exponential prior on it, this has an exponential prior on it. This is written separately for each of those parameters in the core Mr. Bayes code. And so with the more modular way of doing this, you can take that exponential distribution and just use it for whatever is an exponentially distributed parameter. So why does it make sense to actually write it multiple times? So the other thing about the modularity, it makes it easier to extend existing models, to increase the hierarchy where you're able to actually put priors on parameters where you have uncertainty in them. And then we can also, because it enables this flexibility, we can actually also implement different models in the same software and test them in the same framework. Where, for a lot of programs, you can only use the models that they have. When the sky's the limit, you can do whatever you want, really. So RevBayes and Balify, so this is a program that's mostly focused on simultaneous alignment and topology inference, are kind of both using this sort of heavy modularity within the framework. Beast 2 is also a very modular in its design, but this is actually coming from allowing for plugins that are written by developers for new methods and models. But it's actually also where you can still access the core machinery in certain ways. But in any case, this really is a direction that a lot of software is going in scientific computing. So for RevBayes, just some of the details about it. So it's the back end itself is written in C++, which makes it fast and inherently flexible. We use this flexible model specification framework using probabilistic graphical models, and then we have this, sort of hopefully intuitive, easy to learn, interactive language. So just to show an example of the language, so one thing, you think about how you might be able to specify a method or an analysis in a piece of software, and this is always difficult if you look at something like Beast, where everything is in XML, you can kind of see what's going on, but it's so verbose that it's difficult to read. Well, with an interpreted language, as long as you're familiar enough with the language, whoever writes the script, names their variables in reasonable ways, you can actually read and understand what's going on. And so that's really kind of the goal with the language. So this is a Jupyter kernel, so anyone who's familiar with Jupyter notebooks, either through programming in Python or various other kernels that exist. So this is really a way of coupling code snippets with detailed HTML formatted text that will describe what you're doing. So if you think about creating a reproducible pipeline for your analysis that you could share with other researchers, it's pretty clear what you do when you have the markdown formatted text explaining in detail what everything is below. And then we have the GUI. So the GUI is still in development. Right now it works pretty well on only Macs because it was written in Cocoa. And so the idea behind the GUI is this concept of a tool with a palette of sort of a palette with a toolbox. And so you would have this palette and you would assemble your analysis in any logical way. And so in this analysis, we have this tool that reads in the data. This is the alignment tool. And all of the different tools have these outlets and inlets that you actually have to connect to each other. And so this actually puts together this analysis. And so you have the alignment that sends an aligned data matrix to the model tool. This is a model airplane. Like some of the choices in these things I'm not fully on board with, but whatever. It's not my job. But so this is a model airplane representing the model tool. This actually opens up into a palette where you can build a graphical model. This is our MCMC tool that then outputs to a tree summarization tool. So there are actually quite a lot of different other places where this type of philosophy is taken, and so this is kind of a little bit more straightforward way of working with a piece of software that's inherently modular and having a modular analysis like building GUI. So for the rest of the talk, I'm going to kind of get into more of the nitty-gritty about how models are specified in RevBase and kind of how it lends itself to lots of flexibility and things that you can do. So the idea behind RevBase is that there are these components to doing your analysis. So you have your data, you read it in, and you take, based on the data, you will design your graphical model, like a model describing how those data were generated. You fit the model analyze under the model like doing MCMC, and then you output the results. And so the key thing that I'll focus on for the rest of the talk is really that part there. So this modular model design in RevBase is really kind of the important feature of the software, and this really allows us to expand what we want to do, but also enables a framework for doing this joint inference where we can co-estimate all of the things that we have uncertainty in, and so any of our sort of results are really based on accounting for the fact that we don't know with certainty various parts of our analysis. So this is what I'm showing here is a graphical model. It's a very coarse view of one, like zoomed out, where I'm showing that there is some unified process that we might observe, like, we might observe the outcome of, and it's connected to all of these other components of the model. And so in any phylogenetic model, we have a morphological evolution process, or we might have a molecular evolution process and or morphological evolution process. In the stuff that I'm interested in, we have some process that describes how speciation, extinction, and fossil recovery are done, and we might also have a model that describes how continuous traits evolve over the tree. And so given that we have all these components, one of the things that we understand is that they are all connected, because our assumptions based on a lot of these processes is that the underlying species tree, when we're doing a macroevolutionary analysis, is driving the evolution of things like continuous traits and molecular evolution and things like that. And so given that the tree actually can connect all of these processes, then we can unify them in this underlying unified process. So if we zoom into any one of these components, we can actually see what the nuts and bolts are of the graphical model. So this is a probabilistic graphical model in sort of the more zoomed in view, the fine view. And what a graphical model is showing, it's representing all of the assumptions and the conditional dependent structure of the model, to ultimately lay bare transparently, what this model is doing and what it depends on. So all probabilistic graphical models are directed acyclic graphs. So these are graphs where you can actually have cycles in it. So you couldn't have, this node pointing to itself in any way, or also pointing to its parent. And so, like, inherently, any node in this graph has to have, or can have a parent and children, and so that structure actually illuminates how what parameters are dependent on one another. And so this particular model here that I'm showing is basically showing that we have observed a set of sequence data, and they are generated by some underlying Jukes-Cantor model. So this is just where the rate of going from an A to a C is the same as everything else. And it comes from some phylogeny that has a uniform distribution over the topology and has an exponential distribution over the branch lengths. And we can unite the topology and branch lengths into this node here, and that the sequence data are conditionally dependent on the tree with branch lengths and our model of sequence evolution. And so in 2014, we had a paper that this rev base here shouldn't be there, but we had a paper describing probabilistic graphical models for phylogenetics. So if you're interested in this, I definitely recommend reading that. So if we look at this particular model more explicitly, you can see what the different components are. So in a graphical model, we have our observation. So in the case of just doing an unrooted phylogenetic analysis under the Jukes-Cantor model, we have sequence data. We have something that describes our model of substitution, so the JC rate matrix, which is just basically, a rate matrix where everything equals a third. We have a tree with branch lengths, and we assemble that tree from a topology and a set of branch lengths. And so this is our full model, and we could, if we describe this in Rev Base, we could do an analysis under it right away. So there are a lot of components of the graphical model, and the symbolism that we actually chose for this is actually explicit. And so we have, things that, and the various elements denote these different kinds of variables that we have in any hierarchical statistical model. And so we have these constant nodes, and so we specify constant nodes are things that equal to something. And so this is a node that represents a fixed value, or that we know, or that we assert is the truth. And so this could be the value of a prior parameter. We could say, all the branch lengths come from an exponential distribution with that rate of that exponential is equal to 10, because at some point we actually have to make these types of decisions. Or we could say that this might be the dimensions of a parameter. So this is a tree with dimensions of n taxa. So maybe we have 127 taxa, and so that will actually determine the dimensions of the topology that we draw that from. So we also have these stochastic nodes, and these are actually the important ones, and these nodes represent random variables in our model that come from our prior distributions. And so what we say with the stochastic nodes is these nodes are distributed by or distributed according to some distribution, and that these nodes represent these unknown variables that we are then seeking to estimate in our analysis. So this could be something like here, where our branch length parameters are coming from an exponential distribution with parameter lambda. We also have these deterministic nodes. So these nodes are really determined by parent nodes in the model, or they're essentially a function of parent nodes in the model, but they're not actually coming from a distribution. They are unknown, and we do estimate them, but they're only estimated in that every time we update the tree and the branch lengths, we can recompute this tree with branch lengths. So they're essentially just a function or a computation on other nodes in the model. So here we do the tree assembly. So this is actually a function that we have in RevBayes that puts together a set of branch lengths onto a topology. Or we could have another model. Maybe we just are interested in the length of the tree, so the sum of all the branch lengths, where then this would just equal to the sum of every branch length in the tree. So that might be a parameter we might be interested in understanding, looking at the end of our analysis, and we can compute that every time there's an update to the branch lengths in the model. So we also have these observed stochastic nodes. So where we've graded them out, the node actually is a stochastic node just like these are, that it's coming from a distribution, but because we've actually made an observation, we fixed that to a known value. And then this is how actually we can calculate a likelihood, where then we can say, what is the probability of this observation given my model above? And that gives us a way to say that that's a likelihood. So we fix those observed stochastic nodes to our observed data values. We also have these plates. So the plate actually represents just essentially a way of repeating over multiples of the same thing. So it just simplifies the model structure. So if we just take this part of the model, what this part of the model actually does is it expands to this. And so, this you could imagine if for a tree with 127 taxa would be a lot bigger. And so we don't get a lot out of a model when it's represented that way. And so that's why we have something to represent iteration over a number of variables. So that's actually essentially the anatomy of this model, and so once we have these components, you can imagine how you can think about how you might change this if you were doing a different analysis. So we might start with the Jukes-Kanner model, which a lot of people are familiar with. Is just a model that assumes that the rate of going from A to G to A to C and so on is equal. So this is just showing the exchangeability rates of the bases, where each of these arrows is the same width. And so this is just a constant node that we specify as our Q matrix. So our the instantaneous rate matrix of the model is just 1/3 in every single one of those. And so because under this model, we know that that's true, and we don't actually have to estimate any of those parameters because this is a very simple model. And so in RevBayes, if we wanted to specify this, all we would have to do is specify this as a constant node. But we can think about, maybe we want to extend this to the K2P model. And so here we would have change the definition of our model. So the Kimura two-parameter model assumes that transitions happen at a higher rate than transversions, and so you have more frequently going between these states than you do any of the others. So in this case, we have this extra parameter, kappa, that's the transition transversion rate, and this is in this model in our Q matrix. So if we don't know that value, we actually have to, we might want to estimate it in our analysis. And so what we do is specify this is a stochastic node and put some kind of prior. So in this case, this is a gamma distribution centered on 1. And we define this as a constant node is the parameter of that gamma distribution, and we can estimate kappa, and then our Q matrix becomes a deterministic node, because in this function we simply put this value into this matrix. And so if we wanted to specify that in Rev Bayes, we have to specify a constant node for beta, and then we say the node for kappa is distributed according to a gamma distribution with 1, 1. And then our Q matrix is a function that's determined by this function that assembles this Q matrix for the K80 model. We can extend the K80 model to be more complex and be the HKY model, which simply is the same model for the exchangeability rates. But instead we assume that the base frequencies are unequal, or not all the same. And so here we estimate those, we have put a Dirichlet prior on these parameters, and so we have to specify the concentration for the Dirichlet, and the rest of the model is the same. And so here now our function is putting together for an HKY model, given what our state frequencies are and our kappa value is, it can assemble this Q matrix. And then we have something like the general time reversible model, which assumes that all of the exchangeability rates can be different. And so we have now another prior on these rate values that is also coming from a Dirichlet distribution. So now we have to specify the parameters for the Dirichlet for the state frequencies, and then the parameters for the Dirichlet for the rates. And that should say GTR. And so if we had the function GTR here, and we could assemble this, and this should say R, then we have this function that gives us this deterministic node for the Q matrix. So then you can see how the hierarchy and the flexibility enables us to increase the complexity of our models. And so if we just look at this, we can see that you can easily go from a complex model to a simple one and back by just changing the structure of your model. And so obviously this is just a subset of the substitution models that you can do in Rev Bayes, because all of them are possible with this framework. So this really is this molecular evolution process, and so say you want to expand and zoom into that and decide to put the GTR model in there, and so this is actually where we would link up those components of the model. So in my work, I've focused a lot on this part of the model, so models that describe the speciation and extinction process, and also how fossils are sampled back in time. And so this really gets at these models called fossilized birth-death processes that we've been developing in my lab, also in collaboration with Tanya Stadler and many people in her group. And so I just want to talk a little bit at the end about this, and I think I'm getting close to time, so I might just breeze through this a little bit. But so we've been working on ways of integrating fossils into phylogenetics, both as where we might have individual specimen-based samples back in time, or when those fossils are represented by stratigraphic ranges, where the first and last occurrences of a range are all that we have representing those fossils. And so with these types of models, we can integrate these models into analyses where we have also samples from living taxa, and we can account for the underlying phylogeny or we can actually estimate the underlying phylogeny uniting those taxa, or when we only have paleontological data. So many of you who are familiar with work in paleontological fields is for a lot of data sets we might just have first and last occurrences of lineages back in time, and so we can take these data and based on those sampling times and the duration of those ranges, understand dynamics of diversification, origination, and extinction in the fossil record using just those values. So this is all of these models really kind of inherently rely on the structure of the fossilized birth-death process model. So here we're saying that the tree so this is a tree with divergence times and fossils sampled on those, really is conditionally dependent on a speciation rate, an extinction rate, the start origin time of the process, so the time before the first branching event, a rate at which we recover fossils back in time, and a probability of sampling taxa in the present. And so with a lot of the work that we're doing now is we're extending these methods to work with the data from the fossil record. In particular, we're really interested in these paleo data sets where we just have first last occurrence data over these intervals. And so the important thing about these fossilized birth-death methods is that they explicitly account for these incomplete sampling, which a lot of previously described methods don't really allow you to do. We're about to submit a paper describing a simulation study where we took the paleo-based data and compared it to a birth-death model that assumes complete sampling, the boundary crosser method, and this 3-timer method. So these are essentially summary statistic approaches that essentially where the rates are based on the proportions of first and last appearances and intervals in the fossil record. And so these have sort of workarounds for accommodating the sampling issues. And what we've shown with these simulations is that analysis under the fossilized birth-death process, when we're looking at accuracy of estimating speciation, extinction, and fossil sampling, is much more accurate than any of these other methods. And so one thing that this enables us to start to do is more have a better approach for capturing how the data are collected for paleontological data, and also accounting for the underlying, unobservable phylogeny that does exist, but we can integrate those types of things out. And so these types of approaches also allow us to do this co-estimation when we have samples from the present. And this is actually a study I'll talk more about at the Rancho on Friday, even though it's not on plants. And so this kind of brings us to this more holistic approach for doing phylogenetic inference, where we can take a lot of the things that we're interested in doing. So now I've added in the geographic range evolution and co-estimate these under some unified model. And so this is kind of an example of where we're working toward. This is work that we're kind of aspects of which we're all doing now, where we have, so I'm collaborating with Dan Sepka, who's a penguin paleontologist, and so we have continuous, so body size data, lots of fossil data, morphological, molecular data, and we're interested in co-estimating the phylogeny, the divergence times, the relationships of fossil and extant taxa, as well as how body size has changed over time in penguins, and other aspects of their biology. And so with that, I'll just thank my collaborators in lab and others in funding, and thank you guys for listening. Yeah, so we don't have any plans to do that right now. I mean, if that's something you're interested in doing, you're more than welcome to. Some initial attempts at thinking about that, like there were like a while back, there were just some challenges with representing some of the modeling aspects, but I think like with things like RSTAN and others, like that might enable us a way to do it. But yeah, we don't have any plans immediately, just because we think that like a GUI probably is like the easiest entry level interface for RevBase at the moment, and so that's kind of where we're directing a lot of our efforts on that. Yeah.
I was curious about the hope of incorporating uncertainty in the data itself. So let's say you have a bunch of low coverage genomes or something like this.
So that is possible probably already. So we do that with so if you have certain, so in RevBase, the fossil occurrence time is assumed is data. And so we can easily accommodate that by creating a sort of probability distribution according to that represents our uncertainty in the data, and so that's possible for those things. And I could see pretty straightforward ways of doing that with sequence data. It just might take some work. Yeah. Hey.
How's it going?
Good.
Great talk, and I think it's really cool how you can it's so flexible with RevBase. I want to ask you your thoughts. There's this other direction that kind of the Mr. Bays has been packaged up and for efficiency with SBase and those other kind of programs. What are your thoughts about that direction of just going toward efficiency?
So that's actually a big part of this one grant that we have as well. So we're working on sort of MPI versions that will speed up the computation a lot, which will probably only exist for some sort of very standard analyses. Because if you start putting together a super complex model and brand new ways of describing priors on parameters, it's probably not going to be that amenable to MPI like the faster versions. But so I have a new postdoc who's starting in my lab in February who's really interested in developing new MCMC proposals that combine parameters. So one of the biggest challenges with phylogenetics, especially under complex models, is that a lot of these models are really difficult to sample or mix over because of all the correlations and parameters. So you can imagine under a continuous trait evolution model, there might be closely related branches have the same rate of evolution, and so if you want to capture that, any changes you might propose to the tree topology would be easily rejected because they break up that structure, right, that you've settled on in one part of the model. So there's actually quite a lot of different cases where we have these correlated parameters, and so what she's working on is generic ways of coupling correlated parameters to more efficiently estimate over those. So a single one of those moves alone would take more time than, or a combined move takes more time than a single move, but because they're performing them at the same time, it should overall increase the efficiency of the MCMC. So those are really a lot of the things that we're working on. So hopefully we can improve the speed to get to something better for or much better for standard models, and then better for complex ones.
So I have a last question. For some of us who are here interested in particular questions on evolution of traits or biogeography or anything, where should we start if we want to use RevBase, or how people who don't know anything about probabilistic models, where's it going to start, and how do these workshops work? And
So I do think like workshops are a great place to start, but also the workshop materials that we have are all online, but also places like Woods Hole. Like, so some of that, I would say if you don't have a strong background in probability theory or statistics, so Paul Lewis actually did 3 philo seminars this summer where he gave his excellent lectures on sort of probability theory, maximum likelihood, and phylogenetics, and Bayesian phylogenetics. And so they're wonderful, and I definitely recommend starting there. But yeah, like we are trying to get a lot of our tutorials in such a way that they, there's some that are quite difficult because they are complex methods, but hopefully some of the introductory ones start off easy. And we're also open in a way that, like, if users want to create tutorials, that's a possibility through pull requests and stuff like that. So if there are issues with our current tutorials, so that's kind of where I would say get started. But like, with RevBase we have a user forum, and hopefully as that picks up, then like queries and stuff get answered more quickly. Essentially, like, these methods are hard to start and use because they're hard, and like, hopefully there's just enough venues for people to learn them. And so we have workshops where, like, hopefully are going to be, so we're planning 4 workshops, like probably starting sometime next fall, like a year from now, that we hope to be like no cost to very low cost and stuff. And so we're trying to increase the accessibility that way.
So let's thank Tracy again. Remember, 4:30, you want to